Is that a tadpole with massive legs? What evolutionary drama is unfolding here? Well, that's the story of the unsung hero of the animal kingdom, the thyroid hormone. It's often blamed for sluggish metabolism and health woes, but without it, tadpoles would not become frogs, human organs wouldn't know how to grow, and life may have never made the leap from the oceans to the land. Today we're diving deep into why the thyroid hormone was essential for life to flourish on Earth. Please like and subscribe. To fully understand how central the thyroid hormone is, we have to rewind way back. Roughly 3.5 billion years ago, a time when Earth's oceans teemed with only unicellular life forms. These early life forms relied on iodine, a potent antioxidant for survival. In its ionic form, iodide, it could be used to catalyze biological reactions. Over time, it combined with the amino acid tyrosine to form iodotyrosine. Iodotyrosine was a better version than iodine for biological organisms. It was more mobile, reactive, and crucially, it was the precursor for the earliest versions of the thyroid hormone. Fast forward to today, thyroid hormone is a master regulator. In vertebrates, it directs cell signaling, energy production, gene expression, and even DNA replication. So how did this hormone start steering evolution? In many organisms, the availability of iodide became a switch for transitioning between life stages. When iodine is scarce, staying in a juvenile state was a survival advantage, conserving energy and avoiding risky growth, which could not be sustained. But when iodine was abundant, it's time to grow up and pass on those genes. This life stage control was mediated by the thyroid hormone signaling pathway. As an example, let's take the majestic sea urchin. While in their larval form in iodide-poor waters, the thyroid hormone signaling pathway focuses energy on larval feeding structures. These structures would entail more iodine uptake. While in iodide-rich waters, however, the same pathways would redirect energy towards metamorphosis and for the sea urchin larvae to become their majestic final forms. Essentially, in many marine organisms, feeding and iodine uptake became evolutionarily coupled. Eat well to morph well. But nature had a problem. Iodine is concentrated in oceans, but sparse on land. The endostyle evolved in marine chordates, such as tunicates. While primarily a feeding organ, it also functioned as an iodine filter, helping these organisms venture into iodine-poor marine environments or cope with fluctuating iodine availability. Meanwhile, the thyroid gland evolved later in vertebrates, likely from the endostyle, and became a dedicated endocrine organ. It enabled internal iodine storage and regulated thyroid hormone synthesis, which was crucial for life transitioning to land, where iodine was much more scarce. A quick addition here, lampreys are an oddity. In their larval stage, they have an endostyle, and in their adult stage, they have a thyroid gland. This makes them an interesting model to study the evolution of these organs. Around 500 to 600 million years ago, creatures such as the famous Tiktaalik began to crawl onto land. For this transition, a portable iodine regulation system was crucial. For those who developed the endostyle or thyroid gland, they could finally decouple iodine from feeding, gaining freedom to explore new ecosystems. As organisms became more complex, so did their thyroid system. Two key genes emerged, thyroid hormone receptor alpha and thyroid hormone receptor beta. Though structurally similar, they activate very different sets of genes. The evolution of a more advanced thyroid hormone signaling pathway allowed the thyroid hormone to fine-tune development and execute precision across tissues and life stages in many organisms. And nowhere is this clearer than in frogs. In tadpoles, during metamorphosis, thyroid receptor alpha-1 is highly expressed in tissues destined for transformation, such as limb buds. In contrast, thyroid receptor beta surges in tissues that will be reabsorbed, such as gills and the tail. And of course, scientists could not resist the urge to interfere with the tadpole's journey. In the absence of thyroid hormone, thyroid receptor alpha acts as a brake, delaying metamorphosis and promoting growth. Knockout experiments show that by removing thyroid receptor beta prevents tail reabsorption, resulting in frogs with tails. Furthermore, blocking tyrosine receptor alpha-1 in limb buds leads to tadpoles with unusually large legs and bigger bodies. These results show clearly just how central the thyroid hormone is to orchestrating metamorphosis. But this isn't just frog business, it's personal. In humans, failing organs, such as failing heart tissue, often show decreased thyroid 3 levels and increased thyroid receptor alpha 1 level. Administration of thyroid 3 can sometimes restore some function, suggesting the thyroid receptor A1 and thyroid hormone axis is still biologically active in our cells. During regeneration of amputated limbs of lizards and fish, low thyroid 3, high thyroid receptor A1 patterns resemble the same axis we see in failing human organs. Based on this knowledge, could the axial shift we see in failed human organs be linked to an ancient evolutionary stress signal which attempts tissue regeneration, but cannot due to mammals no longer having the ability to complete this process? It's an intriguing theory, but we're far from growing back our limbs. Still, this could hint at a deep evolutionary script waiting to be reactivated. If you enjoyed this video, please like and subscribe, and check out other videos made by this channel. See you next time for more science.